Hi, my name is Lorenzo Caciagli and I'm a clinical research fellow and former PhD candidate at the Department of Clinical and Experimental Epilepsy at the UCL Queen Square Institute of Neurology and at the MRI unit of the Epilepsy Society, Chapel St. Peter, Buckinghamshire.
I'm Matthias Köpf. I'm a consultant neurologist and UCL professor of neurology and had the privilege to supervise Lorenzo over the last 3 years for his PhD studies, and the results of which are now in press.
Today we're going to discuss some of the findings of our recent study, which has just been published in Brain, titled Abnormal Hippocampal Structure and Function in Juvenile Myoclonic Epilepsy and Unaffected Siblings. Juvenile myoclonic epilepsy is the most common genetic generalized epilepsy syndrome and accounts for up to 10% of all epilepsy. It has a complex polygenetic etiology implicating high heritability. Previous investigations detected mesial or lateral frontal cortical abnormalities and impaired frontal-thalamocortical connectivity, and therefore the syndrome has most often been conceptualized as a disorder of frontal-thalamocortical loops. So the presence of mesiotemporal abnormalities and associated memory alterations, on the other hand, is controversial, and studies so far have not tested hippocampal function. So with this study, we aim to shift the focus from the frontal lobe and decided to investigate structure and function of the mesiotemporal lobe in a sample of 37 patients with juvenile myoclonic epilepsy, JME, 16 of their unaffected siblings and 20 healthy controls. And they were all comparable for age, sex, handedness and hemispheric dominance. So we first implemented structural MRI to derive measures of hippocampal volume, and we also tested for markers of atypical hippocampal shape and positioning, which are subsumed under the term of hippocampal malrotation, which refers basically to a hippocampus with a slightly rounded or pyramidal shape, higher overlap of the collateral sulcus and abnormal sulcal morphometry within the parahippocampal gyrus. And these are also to represent neurodevelopmental markers. Okay. In fact, the morphological patterns associated with malrotation were shown to arise from an incomplete inversion of the mesiotemporal structures during gestation. As a functional counterpart, we used neuropsychological tests addressing verbal and visual learning and an event-related functional MRI memory encoding paradigm, including both verbal and visual items. Given the polygenetic background of JME, we investigated both patients and their unaffected siblings in an attempt to identify intermediate phenotypes or endophenotypes, i.e. heritable traits co-segregated in families with affected members and related to disease pathological mechanisms. So we found a reduction of left hippocampal volume, higher frequency of hippocampal malrotation and higher levels of hippocampal verticalization both on the left and the right in patients and their siblings. Quantitative markers of hippocampal malpositioning achieved high level of subgroup discrimination between controls and patients and between controls and a combined group of patients and their unaffected siblings. Though we do not identify overt impairment of verbal or visual learning, we detected abnormal pattern of hippocampal activation, particularly for verbal items, pointing to atypical mesiotemporal and also frontotemporal recruitment, both in patients and their siblings, and with more prominent reductions of frontal and temporal activation, particularly in JME with hippocampal malrotation. Importantly, linear models identified a direct relation between a composite marker of malpositioning and both hippocampal and frontal activation, with more marked hippocampal malpositioning being associated with lower task-related recruitment.
What Lorenzo has shown in collaboration with our neuropsychologists and imaging specialists is very much in line with the current understanding of epilepsy which is the predisposition for seizures associated with its cognitive comorbidities. Here we have an example that epilepsy hijacks a particular system in the brain, the mesiotemporal system, and this predisposition to generate then seizures is also passed through the generations, i.e. is genetically inheritable and seen in siblings of patients who themselves do not suffer from epilepsy. This opens up a whole new realm of possibilities of using epilepsy in those people at risk of developing this condition.